Gene expression is the process by which information from a gene is used in the synthesis of a functional gene product. These products are often proteins, but in non-protein coding genes such as ribosomal RNA, transfer RNA or small nuclear RNA genes, the product is a functional RNA. The process of gene expression is used by all known life, eukaryotes, prokaryotes, and utilized by viruses, to generate the macromolecular machinery for life. Several steps in the gene expression process may be modulated, including the transcription, RNA splicing, translation, and post-translational modification of a protein. Gene regulation gives the cell control over structure and function, and is the basis for cellular differentiation, morphogenesis and the versatility and adaptability of any organism. Gene regulation may also serve as a substrate for evolutionary change, since control of the timing, location, and amount of gene expression can have a profound effect on the functions of the gene in a cell or in a multicellular organism. In genetics, gene expression is the most fundamental level at which the genotype gives rise to the phenotype. The genetic code stored in DNA is interpreted by gene expression, and the properties of the expression give rise to the organism's phenotype. Such phenotypes are often expressed by the synthesis of proteins that control the organism's shape, or that act as enzymes catalyzing specific metabolic pathways characterizing the organism. Mechanism, transcription. A gene is a stretch of DNA that encodes information. Genomic DNA consists of two antiparallel and reverse complementary strands, each having 5' and 3' ends. With respect to a gene, the two strands may be labeled the template strand, which serves as a blueprint for the production of an RNA transcript, and the coding strand, which includes the DNA version of the transcript sequence. The production of RNA copies of the DNA is called transcription, and is performed in the nucleus by RNA polymerase, which adds one RNA nucleotide at a time to a growing RNA strand. This RNA is complementary to the template 3' to 5 DNA strand, which is itself complementary to the coding 5' to 3 DNA strand. Therefore, the resulting 5' to 3 RNA strand is identical to the coding DNA strand with the exception that thymines are replaced with uracils in the RNA. A coding DNA strand reading ATG is indirectly transcribed through the non-coding strand as AUG in RNA. Transcription in prokaryotes is carried out by a single type of RNA polymerase, which needs a DNA sequence called a pribno box as well as a sigma factor to start transcription. In eukaryotes, transcription is performed by three types of RNA polymerases, each of which needs a special DNA sequence called a promoter and a set of DNA-binding proteins, transcription factors to initiate the process. RNA polymerase I is responsible for transcription of ribosomal RNA genes. RNA polymerase II transcribes all protein-coding genes but also some non-coding RNAs. Pol2 includes a C-terminal domain that is rich in serine residues. When these residues are phosphorylated, the CTD binds to various protein factors that promote transcript maturation and modification. RNA polymerase III transcribes 5 srRNA, transfer RNA genes, and some small non-coding RNAs. Transcription ends when the polymerase encounters a sequence called the terminator. RNA processing. While transcription of prokaryotic protein-coding genes creates messenger RNA that is ready for translation into protein, transcription of eukaryotic genes leaves a primary transcript of RNA, which first has to undergo a series of modifications to become a mature mRNA. These include 5' capping, which is a set of enzymatic reactions that add 7-methylguanosine to the 5' end of pre-mRNA and thus protect the RNA from degradation by exonucleases. The M7G cap is then bound by cap-binding complex heterodimer, which aids in mRNA export to cytoplasm and also protect the RNA from decoupling. Another modification is 3' cleavage and polyadenylation. They occur if polyadenylation signal sequence is present in pre-mRNA, which is usually between protein coding sequence and terminator. The pre-mRNA is first cleaved and then a series of 200 adenines are added to form poly, A, tail, which protects the RNA from degradation. Poly-A tail is bound by multiple poly-A binding proteins necessary for mRNA export and translation reinitiation. A very important modification of eukaryotic pre-mRNA is RNA splicing. The majority of eukaryotic pre-mRNAs consist of alternating segments called exons and introns. During the process of splicing, an RNA protein catalytical complex known as spliceosome catalyzes two transesterification reactions, which remove an intron and release it in form of lariat structure, and then splice neighboring exons together. In certain cases, some introns or exons can be either removed or retained in mature mRNA. This so-called alternative splicing creates series of different transcripts originating from a single gene. Because these transcripts can be potentially translated into different proteins, splicing extends the complexity of eukaryotic gene expression. Extensive RNA processing may be an evolutionary advantage made possible by the nucleus of eukaryotes. In prokaryotes, transcription and translation happen together, whilst in eukaryotes the nuclear membrane separates the two processes, giving time for RNA processing to occur. Non-coding RNA maturation. In most organisms, non-coding genes are transcribed as precursors that undergo further processing. In the case of ribosomal RNAs, they are often transcribed as a pre-rRNA that contains one or more rRNAs. The pre-rRNA is cleaved and modified at specific sites by approximately 150 different small nucleolus-restricted RNA species, called snRNAs. snRNAs associate with proteins, forming snRNPs. While snRNA part base pair with a target RNA and thus position the modification at a precise site, the protein part performs the catalytical reaction. In eukaryotes, in particular a snRNP called RNase, MRP cleaves the 45S pre-rRNA into the 28S, 5.8S, and 18S rRNAs. The rRNA and RNA processing factors form large aggregates called the nucleolus. In the case of transfer RNA, for example, the 5' sequence is removed by RNase-P, whereas the 3' end is removed by the tRNase-Z enzyme, and the non-templated 3' CCA tail is added by a nucleotidal transferase. In the case of microRNA, miRNAs are first transcribed as primary transcripts or prime-miRNA with a cap and poly-A tail and processed to short, 70-nucleotide stem loop structures known as pre-miRNA in the cell nucleus by the enzymes DROSHA and PASHA. After being exported, it is then processed to mature miRNAs in the cytoplasm by interaction with the endonuclease dicer, which also initiates the formation of the RNA-induced silencing complex, composed of the Argonaute protein. Even snRNAs and snRNAs themselves undergo series of modification before they become part of functional RNP complex. This is done either in the nucleoplasm or in the specialized compartments called cagal bodies. Their bases are methylated or pseudoadenylated by a group of small cagal body-specific RNAs, which are structurally similar to snRNAs. RNA export. In eukaryotes, most mature RNA must be exported to the cytoplasm from the nucleus. While some RNAs function in the nucleus, many RNAs are transported through the nuclear pores and into the cytosol. Notably, this includes all RNA types involved in protein synthesis. In some cases, RNAs are additionally transported to a specific part of the cytoplasm, such as a synapse. They are then towed by motor proteins that bind through linker proteins to specific sequences on the RNA. Translation. For some RNA, the mature RNA is the final gene product. In the case of messenger RNA, the RNA is an information carrier coding for the synthesis of one or more proteins. mRNA carrying a single protein sequence is monocistronic, whilst mRNA carrying multiple protein sequences is known as polycistronic. Every mRNA consists of 3 parts: 5' untranslated region, protein coding region or open reading frame, and 3' untranslated region. The coding region carries information for protein synthesis encoded by the genetic code to form triplets. Each triplet of nucleotides of the coding region is called a codon and corresponds to a binding site complementary to an anticodon triplet in transfer RNA. Transfer RNAs with the same anticodon sequence always carry an identical type of amino acid. Amino acids are then chained together by the ribosome according to the order of triplets in the coding region. The ribosome helps transfer RNA to bind to messenger RNA and takes the amino acid from each transfer RNA and makes a structure-less protein out of it. Each mRNA molecule is translated into many protein molecules, on average 2,800 in mammals. In prokaryotes, translation generally occurs at the point of transcription, often using a messenger RNA that is still in the process of being created. In eukaryotes, translation can occur in a variety of regions of the cell depending on where the protein being written is supposed to be. Major locations are the cytoplasm for soluble cytoplasmic proteins and the membrane of the endoplasmic reticulum for proteins that are for export from the cell or insertion into a cell membrane. Proteins supposed to express at the endoplasmic reticulum are recognized partway through the translation process. This is governed by the signal recognition particle a protein that binds to the ribosome and directs it to the endoplasmic reticulum when it finds a signal peptide on the growing amino acid chain. Translation is the communication of the meaning of a source language text by means of an equivalent target language text, folding. The polypeptide folds into its characteristic and functional three-dimensional structure from a random coil. Each protein exists as an unfolded polypeptide or random coil when translated from a sequence of mRNA into a linear chain of amino acids. This polypeptide lacks any developed three-dimensional structure. Amino acids interact with each other to produce a well-defined three-dimensional structure, the folded protein known as the native state. The resulting three-dimensional structure is determined by the amino acid sequence. The correct three-dimensional structure is essential to function, although some parts of functional proteins may remain unfolded. Failure to fold into the intended shape usually produces inactive proteins with different properties including toxic prions. Several neurodegenerative and other diseases are believed to result from the accumulation of misfolded proteins. Many allergies are caused by the folding of the proteins, for the immune system does not produce antibodies for certain protein structures. Enzymes called chaperones assist the newly formed protein to attain the three-dimensional structure it needs to function. Similarly, RNA chaperones help RNAs attain their functional shapes. Assisting protein folding is one of the main roles of the endoplasmic reticulum in eukaryotes. Translocation. Secretory proteins of eukaryotes or prokaryotes must be translocated to enter the secretory pathway. Newly synthesized proteins are directed to the eukaryotic Sec61 or prokaryotic Sec-YEG translocation channel by signal peptides. Whereas the efficiency of protein secretion in eukaryotes is very dependent on the signal peptide who has been used. Protein transport. Many proteins are destined for other parts of the cell than the cytosol, and a wide range of signaling sequences are used to direct proteins to where they are supposed to be. In prokaryotes this is normally a simple process due to limited compartmentalization of the cell. However, in eukaryotes there is a great variety of different targeting processes to ensure the protein arrives at the correct organelle. Not all proteins remain within the cell and many are exported, for example digestive enzymes, hormones and extracellular matrix proteins. In eukaryotes the export pathway is well developed and the main mechanism for the export of these proteins is translocation to the endoplasmic reticulum, followed by transport via the Golgi apparatus. Regulation of gene expression. Regulation of gene expression refers to the control of the amount and timing of appearance of the functional product of a gene. Control of expression is vital to allow a cell to produce the gene products it needs when it needs them. In turn, this gives cells the flexibility to adapt to a variable environment, external signals, damage to the cell, etc. Some simple examples of where gene expression is important are, control of insulin expression so it gives a signal for blood glucose regulation, X chromosome inactivation in female mammals to prevent an overdose of the genes it contains. Cyclin expression levels control progression through the eukaryotic cell cycle. More generally, gene regulation gives the cell control over all structure and function, and is the basis for cellular differentiation, morphogenesis and the versatility and adaptability of any organism. Any step of gene expression may be modulated, from the DNA-RNA transcription step to post-translational modification of a protein. The stability of the final gene product, whether it is RNA or protein, also contributes to the expression level of the gene. An unstable product results in a low expression level. In general, gene expression is regulated through changes in the number and type of interactions between molecules that collectively influence transcription of DNA and translation of RNA. Numerous terms are used to describe types of genes depending on how they are regulated. These include: a constitutive gene is a gene that is transcribed continually as opposed to a facultative gene, which is only transcribed when needed. A housekeeping gene is typically a constitutive gene that is transcribed at a relatively constant level. The housekeeping gene's products are typically needed for maintenance of the cell. It is generally assumed that their expression is unaffected by experimental conditions. Examples include actin, GAPDH and ubiquitin. A facultative gene is a gene only transcribed when needed as opposed to a constitutive gene. An inducible gene is a gene whose expression is either responsive to environmental change or dependent on the position in the cell cycle. Transcriptional regulation. Regulation of transcription can be broken down into three main routes of influence. Genetic, modulation interaction of a control factor with the transcription machinery, and epigenetic, non-sequence changes in DNA structure that influence transcription. Direct interaction with DNA is the simplest and the most direct method by which a protein changes transcription levels. Genes often have several protein binding sites around the coding region with the specific function of regulating transcription. There are many classes of regulatory DNA binding sites known as enhancers, insulators and silencers. The mechanisms for regulating transcription are very varied, from blocking key binding sites on the DNA for RNA polymerase to acting as an activator and promoting transcription by assisting RNA polymerase binding. The activity of transcription factors is further modulated by intracellular signals causing protein post-translational modification, including phosphorylated, acetylated, or glycosylated. These changes influence a transcription factor's ability to bind, directly or indirectly, to promote a DNA, to recruit RNA polymerase, or to favor elongation of a newly synthesized RNA molecule. The nuclear membrane in eukaryotes allows further regulation of transcription factors by the duration of their presence in the nucleus, which is regulated by reversible changes in their structure and by binding of other proteins. Environmental stimuli or endocrine signals may cause modification of regulatory proteins, eliciting cascades of intracellular signals, which result in regulation of gene expression. More recently, it has become apparent that there is a huge influence of non-DNA sequence-specific effects on translation. These effects are referred to as epigenetic and involve the higher-order structure of DNA, non-sequence-specific DNA-binding proteins and chemical modification of DNA. In general, epigenetic effects alter the accessibility of DNA to proteins and so modulate transcription. DNA methylation is a widespread mechanism for epigenetic influence on gene expression and is seen in bacteria and eukaryotes and has roles in heritable transcription silencing and transcription regulation. In eukaryotes, the structure of chromatin, controlled by the histone code, regulates access to DNA with significant impacts on the expression of genes in euchromatin and heterochromatin areas. Post-transcriptional regulation. In eukaryotes, where export of RNA is required before translation is possible, nuclear export is thought to provide additional control over gene expression. All transport in and out of the nucleus is via the nuclear pore, and transport is controlled by a wide range of importin and exportin proteins. Expression of a gene coding for a protein is only possible if the messenger RNA carrying the code survives long enough to be translated. In a typical cell, an RNA molecule is only stable if specifically protected from degradation. RNA degradation has particular importance in regulation of expression in eukaryotic cells where mRNA has to travel significant distances before being translated. In eukaryotes, RNA is stabilized by certain post-transcriptional modifications, particularly the 5' cap and polyadenylated tail. Intentional degradation of mRNA is used not just as a defense mechanism from foreign RNA but also as a route of mRNA destabilization. If an mRNA molecule has a complementary sequence to a small interfering RNA, then it is targeted for destruction via the RNA interference pathway. Translational regulation. Direct regulation of translation is less prevalent than control of transcription or mRNA stability but is occasionally used. Inhibition of protein translation is a major target for toxins and antibiotics, so they can kill a cell by overriding its normal gene expression control. Protein synthesis inhibitors include the antibiotic neomycin and the toxin ricin. Protein degradation. Once protein synthesis is complete, the level of expression of that protein can be reduced by protein degradation. There are major protein degradation pathways in all prokaryotes and eukaryotes, of which the proteasome is a common component. An unneeded or damaged protein is often labeled for degradation by addition of ubiquitin. Measurement. Measuring gene expression is an important part of many life sciences. The ability to quantify the level at which a particular gene is expressed within a cell, tissue or organism can give a huge amount of information. For example, measuring gene expression can identify viral infection of a cell, determine an individual's susceptibility to cancer, find if a bacterium is resistant to penicillin. Similarly, the analysis of the location of expression protein is a powerful tool, and this can be done on an organism or cellular scale. Investigation of localization is particularly important for study of development in multicellular organisms and is an indicator of protein function in single cells. Ideally, measurement of expression is done by detecting the final gene product. However, it is often easier to detect one of the precursors, typically mRNA, and inferred gene expression level. mRNA quantification. Levels of mRNA can be quantitatively measured by Northern blotting, which provides size and sequence information about the mRNA molecules. A sample of RNA is separated on an agarose gel and hybridized to a radioactively labeled RNA probe that is complementary to the target sequence. The radiolabeled RNA is then detected by an autoradiograph. Because the use of radioactive reagents makes the procedure time-consuming and potentially dangerous, alternative labeling and detection methods, such as digoxygenin and biotin chemistries, have been developed. Perceived disadvantages of Northern blotting are that large quantities of RNA are required and that quantification may not be completely accurate, as it involves measuring band strength in an image of a gel. On the other hand, the additional mRNA size information from the Northern blot allows the discrimination of alternately spliced transcripts. Another approach for measuring mRNA abundance is RT-qPCR. In this technique, reverse transcription is followed by quantitative PCR. Reverse transcription first generates a DNA template from the mRNA. This single-stranded template is called cDNA. The cDNA template is then amplified in the quantitative step, during which the fluorescence emitted by labeled hybridization probes or intercalating dyes changes as the DNA amplification process progresses. With a carefully constructed standard curve, qPCR can produce an absolute measurement of the number of copies of original mRNA, typically in units of copies per nanoliter of homogenized tissue or copies per cell. qPCR is very sensitive, but can be expensive depending on the type of reporter used. Fluorescently labeled oligonucleotide probes are more expensive than non-specific intercalating fluorescent dyes. For expression profiling, or high-throughput analysis of many genes within a sample, quantitative PCR may be performed for hundreds of genes simultaneously in the case of low-density arrays. A second approach is the hybridization microarray. A single array or chip may contain probes to determine transcript levels for every known gene in the genome of one or more organisms. Alternatively, tag-based technologies like serial analysis of gene expression and RNA-seq, which can provide a relative measure of the cellular concentration of different mRNAs, can be used. An advantage of tag-based methods is the open architecture, allowing for the exact measurement of any transcript, with a known or unknown sequence. Next-generation sequencing such as RNA-seq is another approach, producing vast quantities of sequence data that can be matched to a reference genome. Although NGS is comparatively time-consuming, expensive, and resource-intensive, it can identify single nucleotide polymorphisms, splice variants, and novel genes, and can also be used to profile expression in organisms for which little or no sequence information is available. Protein quantification. For genes encoding proteins, the expression level can be directly assessed by a number of means with some clear analogies to the techniques for mRNA quantification. The most commonly used method is to perform a Western blot against the protein of interest. This gives information on the size of the protein in addition to its identity. A sample is separated on a polyacrylamide gel, transferred to a membrane and then probed with an antibody to the protein of interest. The antibody can either be conjugated to a fluorophore or to horseradish peroxidase for imaging and/or quantification. The gel-based nature of this assay makes quantification less accurate, but it has the advantage of being able to identify later modifications to the protein, for example proteolysis or ubiquitination, from changes in size. Localization. Analysis of expression is not limited to only quantification. Localization can also be determined. mRNA can be detected with a suitably labeled complementary mRNA strand, and protein can be detected via labeled antibodies. The probed sample is then observed by microscopy to identify where the mRNA or protein is. By replacing the gene with a new version fused to a green fluorescent protein marker, expression may be directly quantified in live cells. This is done by imaging using a fluorescence microscope. It is very difficult to clone a GFP-fused protein into its native location in the genome without affecting expression levels, so this method often cannot be used to measure endogenous gene expression. It is, however, widely used to measure the expression of a gene artificially introduced into the cell, for example via an expression vector. It is important to note that by fusing a target protein to a fluorescent reporter, the protein's behavior, including its cellular localization and expression level, can be significantly changed. The enzyme-linked immunosorbent assay works by using antibodies immobilized on a microtiter plate to capture proteins of interest from samples added to the well. Using a detection antibody conjugated to an enzyme or fluorophore, the quantity of bound protein can be accurately measured by fluorometric or colorimetric detection. The detection process is very similar to that of a Western blot, but by avoiding the gel steps, more accurate quantification can be achieved. Expression system. An expression system is a system specifically designed for the production of a gene product of choice. This is normally a protein, although may also be RNA, such as tRNA or a ribozyme. An expression system consists of a gene, normally encoded by DNA, and the molecular machinery required to transcribe the DNA into mRNA and translate the mRNA into protein using the reagents provided. In the broadest sense, this includes every living cell, but the term is more normally used to refer to expression as a laboratory tool. An expression system is therefore often artificial in some manner. Expression systems are, however, a fundamentally natural process. Viruses are an excellent example where they replicate by using the host cell as an expression system for the viral proteins and genome. Inducible expression, doxycycline is also used in TET-on, and TET-off tetracycline-controlled transcriptional activation to regulate transgene expression in organisms and cell cultures. The filamentous fungus Mycelia thermophila, C1.1, was developed into an expression platform for screening and production of diverse industrial enzymes. C1 shows a low viscosity morphology in submerged culture, enabling the use of complex growth and production media. The commercially relevant expression system based on Mycelia thermophila, C1, was sequenced by the Joint Genome Institute, 2, and Dyadic International, Incorporated, 3. It has been licensed for the commercial production of enzymes and other proteins by internationally recognized companies such as BASF, Abingo Bioenergy, Sanofi Pasteur, Codexis, and others. In nature, in addition to these biological tools, Certain naturally observed configurations of DNA and the associated machinery itself are referred to as an expression system. This term is normally used in the case where a gene or set of genes is switched on under well-defined conditions. For example, the simple repressor switch expression system in lambdaphage and the lac operator system in bacteria. Several natural expression systems are directly used or modified and used for artificial expression systems such as the tet-on and tet-off expression system. Gene networks. Genes have sometimes been regarded as nodes in a network, with inputs being proteins such as transcription factors, and outputs being the level of gene expression. The node itself performs a function, and the operation of these functions have been interpreted as performing a kind of information processing within cells and determine cellular behavior. Gene networks can also be constructed without formulating an explicit causal model. This is often the case when assembling networks from large expression data sets. Covariation and correlation of expression is computed across a large sample of cases and measurements. The source of variation can be either experimental or natural. There are several ways to construct gene expression networks, but one common approach is to compute a matrix of all pairwise correlations of expression across conditions, time points, or individuals and convert the matrix into a graphical representation in which nodes represent genes, transcripts, or proteins and edges connecting these nodes represent the strength of association. Weighted correlation network analysis involves weighted networks defined by soft thresholding the pairwise correlations among variables. Techniques and tools. The following experimental techniques are used to measure gene expression and are listed in roughly chronological order, starting with the older, more established technologies. They are divided into two groups based on their degree of multiplexity. Low to mid-plex techniques, reporter gene, northern blot, western blot, fluorescent in situ hybridization, reverse transcription PCR. Higher plex techniques, SAGE, DNA microarray, tiling array, RNA-seq. See also, allomap molecular expression testing, bookmarking, epigenetics, expressed sequence tag, expression profiling, genetic engineering, genetically modified organism, list of human genes, oscillating gene, paramutation, ribonomics, ridges, sequence profiling tool, transcriptional bursting, transcriptional noise, references. External links, genes and gene expression. The Virtual Library of Biochemistry and Cell Biology. Biochemweb.org December 4, 2005. Retrieved June 10, 2008. John Crick. DNA makes RNA. Retrieved June 10, 2008. Advancing Gene Expression Studies. Genetic Engineering and Biotechnology News. Mary Ann Liebert, Incorporated August 1, 2008. Optimizing Transient Gene Expression. Genetic Engineering and Biotechnology News. Mary Ann Liebert, Incorporated March 1, 2008. 5, 6.